The overall goal of this procedure is to develop an in vitro model to study the effects of Plasmodium falciparum-infected erythrocytes on the replication of HIV-1 in human primary monocyte-derived macrophages. This is accomplished by first isolating Plasmodium falciparum-infected red blood cells using the VarioMax system and then exposing human monocyte-derived macrophages to the infected cells. Next, the red blood cell exposed monocyte-derived macrophages are infected with fully replicative HIV or single replication luciferase encoding HIV. Finally, the culture supernatants are harvested at days 3, 6, 9, and 12 post-infection for the fully replicative virus. For the luciferase encoding virus, the infected cells are lysed after 72 hours of infection. Ultimately, viral replication can be monitored by quantifying the amount of HIV-1 p24 capsid protein in supernatants by ELISA, and viral transcription can be monitored by luciferase quantitation of the cell lysates.
This method can help answer key questions in the field of malaria-HIV co-infections. For example, how does infection with the Plasmodium parasite influences HIV replication? Though this method was developed to study interactions between Plasmodium and HIV-1 in macrophages, it could also be applied to other cell types, for example, dendritic cells, monocytes, or CD4 T cells. Demonstrating the procedure will be Guadalupe Andreani, a postdoctoral fellow in the lab.
Begin by harvesting red blood cells from the culture plate.